Yes, students, let us start with a new chapter, cell cycle and cell division. You are aware that all organisms, from the simplest prokaryotic unicellular organism to the giant multicellular organism, start their life from a single cell. You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells. Each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population, which is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division which allow a single cell to form a structure consisting of millions of cells. Now, cell division is a very important process in all living organisms. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two new daughter cells is termed cell cycle. Although cell cycle, in terms of cytoplasmic increase, is a continuous process, the DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. The phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism. Each cell divides every 90 minutes. The cell cycle is divided into two basic phases, interphase and M phase or mitotic phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive mitotic division phases or M phases. The M phase starts with the nuclear division and corresponds to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for cell division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, S phase, and G2 phase. The G1 phase corresponds to the interval between mitosis and initiation of DNA replication. The G1 phase in the cell is metabolically active and continuously grows, but does not replicate its DNA. In animal cells, during the S phase, DNA replication begins in the nucleus and centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in adult animals do not appear to exhibit cell division, and many other cells divide only occasionally as needed to replace the cells that have been lost during injury or cell death. These cells do not enter the G1 phase, but enter into an inactive stage called the quiescent stage or the G0 stage of the cell division. Cells in this stage remain metabolically active but no longer proliferate unless on to do so depending on the requirement of the organism. The M phase or karyokinesis phase is the most dramatic period of the cell cycle. The number of chromosomes in the parent and the progeny is same in mitotic division and thus is called the equational division. Though for convenience we have divided the mitosis into 4 different stages, but all these are progressive in nature and very clear-cut lines cannot be drawn between the stages. The karyokinesis involves prophase, metaphase, anaphase and telophase. Involves prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The characteristic feature of prophase is compact mitotic chromosomes are formed and mitotic spindles are formed. Thus, chromosomal condensation to form compact mitotic chromosomes are seen. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. At the end of prophase, the following structures cannot be seen: the nucleolus, mitochondria and endoplasmic reticulum. The prophase leads to metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, the condensation of chromosome is completed and they are observed clearly under microscope. The characteristic feature of metaphase are spindle fibers attached to the kinetochores of the chromosomes, and chromosomes move to the spindle equator and align themselves at the metaphase plate. The third is anaphase. At this stage, the centromere splits and the chromatid separates. They start moving towards the pole. The chromatids, they lie opposite to each other. The centromere faces the pole. The next is the telophase. Telophase is marked by the chromosome clusters at the opposite spindle poles. The chromosomes lose individuality as discrete elements. The nuclear envelope is formed. And endoplasmic reticulum, nucleolus, Golgi complex reappears. Followed telophase is the cytokinesis phase. The mitosis along with the cytokinesis phase is known as the M phase. At the end of mitosis, 2 daughter nuclei or 2 daughter cells are formed, followed by the cytokinesis. In the stage of cytokinesis, the cell membrane divides. In animal cell, the cell membrane divides by furrow formation. But in plant cells which have a rigid cell wall, the Cell membrane is formed by cell plate formation in the cytoplasm, which is actually the middle lamella of the plant cell. The mitochondria and the plastids are equally redistributed. Now, in some cells, the division of nucleus is not followed by the division of the cytoplasm, and cell membranes are not formed, as in the case of embryonic stage of Drosophila and the liquid endosperm of coconut. Here, this is known as the syncytium. Now, next is the significance of mitosis. Mitosis is responsible for the growth of the organism, leading to the formation of the multicellular organisms and the replacement of the lost part. In plant cell, the mitosis is seen in the apical and the lateral meristematic tissues, which leads to the growth of the tissues in those plants. In animals, it is not only responsible for the growth of the animal, but it is also the process for the replacement of the lost blood cells, epidermal cells, and the gut cells. Blood cells, epidermal cells, and the gut